Well, welcome everybody. I am so excited to have you all here today at our Research to Innovation Showcase. At BIO5, we bring people together. That's one of the main things we do, and have those exciting serendipitous type collisions where people from different areas of research can come together and find new opportunities at the interfaces. And so through BIO5, we help bring people together, and we help fund that in various ways through the seed grants, through equipment grants, through the COVID-19 rapid response grants. That was a special one that we ran this year. And this year it's been a little bit odd, right? People have gotten together or worked from home a lot, worked virtually. We're getting used to seeing each other in all these little squares, but we've continued to go. And one of the really special things, and thank you so much, Michael Johnson, for spearheading this effort, is a way to help promote the professional development of our postdocs, to give you a named fellowship, to allow you to get together a mentoring team, to do some really amazing work, maybe have a little bit of money to go, well, in normal years, travel, at least in this case, engage in some professional development activities, and hopefully get back to normal and go travel in the future. And so now we're going to get to hear about the great work that all of the BIO5 postdoctoral fellows have been able to do. And I am really looking forward to hearing all about it myself. So without further ado, let's get started.
Hi, everyone. I'm so happy that I had the opportunity to take this on. I was very fortunate that Jennifer was very generous with the funds that we could kind of run this particular program. This is the third cohort, and I think we keep getting better. The applications keep getting more and more detailed, and I think that it's really starting to build momentum to really help the postdocs here apply to more for more funding, and to be a lot more, it leads them to even better careers. And we're just really, really happy to be able to support that, because, of course, now we need that mentorship more than anything, because we're so separated right now. But, hopefully things will get back to normal quite soon. So without further ado, I can stop babbling, and I can actually start saying what you came here to hear, and that's hear our great postdocs give a talk. So the first speaker we have is actually Rob Jackson, and he will be talking to us about HPV. So take it away, Rob.
Okay, there. I think I'm unmuted now. Sorry, my slides are just giving me a little problem here. All right, thank you very much for the introduction, Dr. Johnson. I'm very pleased and grateful today to be talking about the BIO5 postdoc fellowship project, which I'm undertaking. I'm from the Van Doorslayer lab, and we study human papillomaviruses. And I'm going to talk about how we decipher HPV infection in lab-grown three-dimensional tissues. So human papillomaviruses are a family of viruses that can cause cancer. About 5% of all cancers worldwide are caused by human papillomaviruses. Now, there's hundreds of different types of this virus, some of them causing warts, some of them causing cancers. The two most common that are causing cancers are HPV type 16 and type 18, and these cause oral as well as anogenital cancers. And an interesting fact in the United States, as well as countries like Canada, is that oral cancers caused by HPV, so that's cancers in the mouth and the back of the throat, like in the tonsils or the base of the tongue, these are outpacing the current rate of cervical cancers that are caused by HPV. Now, biologically speaking, an interesting point of these oral cancers is that nearly 100% of them are caused by HPV type 16 in particular. Now, when we think about HPV infection and how that may lead to cancer, we need to think about the context of the tissue that's infected by HPV. HPV infects epithelial tissues. These are the tissues that make up the skin, the mucosal membranes that line our entire body. HPV can enter the cells in this bottom layer of our epithelium and cause those cells to continue proliferating or growing and dividing to make more viral copies. And over time, as these cells are changing into these upper layer cells, we call this process of change differentiation, the virus life cycle is completed and infectious viral particles can then be shed and the virus life cycle can continue. Now, cancer caused by HPV is really caused by a persistence or a long-term infection that is persisting over years, decades perhaps, and causing these changes in these cells, which eventually can lead to cancer. So when we're thinking about HPV's life cycle, what it's doing to these host cells, we need to consider that HPV's life cycle is differentiation dependent. It absolutely requires that 3D context, which means that when we're studying it in the lab, we need to study it in 3 dimensions. So this final stage of the viral life cycle, what we call productive amplification, where the virus is packaged into this protein shell, that requires 3 dimensions. So we have different models that we can use or make in the lab to study HPV's full viral life cycle. One of the techniques that I'm very familiar with is organotypic raft culturing. And this is just a cross section here of tissue, which we've grown in the lab using this technique. Now we call it raft culture because we're floating these tissue pieces that we're growing in the lab on top of a membrane on a liquid interface, and they're exposed to the air. Now, an alternate technique, which is the subject of the BIO5 postdoc fellowship project, which I've proposed, is to grow these sphere aggregates using bioreactors. So I'll talk a little bit about that. But first, I want to talk about why we use raft cultures at all. They're a relevant model for studying HPV's life cycle. We can see here on the left, this is a cross-section image of normal cervical epithelium, which we've grown in the lab from cervical cells. And on the right is HPV-18 infected cervical cells. And we can see evidence of HPV's impact on this tissue. It's causing increase in proliferation, all these extra cells, these nuclei building up in this compartment of the skin. We can also see these atypical nuclei, which is a sign of the stress that HPV is putting on these tissues. Another interesting aspect of these raft culture models is that it's origin specific. What I mean by this is we can take donor cells from patient samples from tonsils, cervix, foreskin, and these will recapitulate or be able to regrow into the structures that we see in those original tissues. Now, why would we want to use a different approach? So a complementary method like bioreactors allows us to overcome some of the hurdles of raft cultures. Raft cultures are labor intensive. They're not easily amenable to high throughput experiments. So in a collaboration with Dr. Melissa Herbst-Karlowitz's lab at the Phoenix campus, we're growing these bioreactor aggregates. These are functional. They act like real tissue. They secrete the types of proteins that real tissue is secreting in that 3D context. And as well, they provide ample material from this bioreactor for downstream work. So can we use bioreactors to study HPV as a subject of the BIO5 postdoc fellowship? What I'm doing is I'm taking those oral cells, growing them in 3D aggregates and confirming that they can differentiate and become 3D and support a viral life cycle. I've already completed one trial, but ultimately we want to understand how HPV-16 interacts with these host processes in oral epithelium and try to understand more about the biology that's underlying why HPV-16 in particular is causing nearly all these oral cancers, which are on the rise. So I just want to thank you very much for listening to my data blitz. If you'd like to learn more, please visit the poster. It's up on the VoiceThread link, which you should have if you registered. And I will be in a breakout room after to answer questions as well. Thank you very much.
Thanks for that talk, Rob. And as he said, in lieu of asking questions right after each of the talks, what we're going to do is if you go to the breakout rooms, we'll have all of our speakers go to breakout rooms right after this, and you can go there and ask questions. We'll have it open for maybe about 10 minutes. That way, if you want to, ask Rob a question, and then you want to ask our next speaker, Morgan, a question, you'll be able to do that. So feel free, write them down so that you don't forget them, and you can ask all the questions you want, well, hopefully within that 10-minute time frame. So our next speaker is Dr. Morgan Carter, and she's going to talk to us about how do you live inside of a fungi or fungus. So I'm curious to hear about this one as well. Take it away.
Okay, hi everybody. So I'm coming to you from the Baltris lab in collaboration with Betsy Arnold's lab over in the School of Plant Sciences. And my research question that I'm trying to think about might seem very simple on the surface. It's how do you live inside a fungus? And to get at that question, you first have to ask, well, what can live inside of a fungus? So endohyphobacteria, or EHB, are bacteria that live inside of fungal tissues. They were first identified in arbuscular mycorrhizal fungi in the 1970s and 80s. So that's plant-associated fungi. But in the past decade or so, diverse EHB have been found across tons of fungi, plant pathogens, animal pathogens, symbionts, just a huge mixture. And this includes facultative bacterial partners of endophytic fungi, so fungi that inhabit plants in a positive or kind of flat way. But we don't really know much about these. Like I said, they were just discovered in the past decade or so. So the molecular mechanisms underlying them are largely unknown, and the field is really still emerging as to how and when and why and where these relationships form. But what we do already know from the relationships that we've seen so far is that they can have really big impacts, not only on their fungal host, but then on an animal or plant host after that, especially in plants where we see that the fungi that have bacteria may have different ability to reproduce, may produce different toxins or different levels of plant hormones. So there's a lot to be said about learning more about these interactions to be able to then use them to learn more about these systems as well as to manipulate them. So my research question in coming to the University of Arizona was really, how do bacteria invade fungi and establish symbiosis? And specifically, what genes are required for or involved in partnership? So on this slide, I'm showing you a photo of the study system that I'm using, where you can see little red bacteria of Lutibacter microvicinus are hanging out inside of a blue hyphae of Phthalates fungus, which is a tree endophyte. So to ask what genes are required for partnership, I'm going about this in two ways. One is a little more conventional, and that's just RNA-seq analysis. So we can do dual RNA-seq, where we can look at gene expression changes in the bacterium and in the fungus when they're co-cultured versus when they're cultured independently, to look at what's turned on and off when you're interacting with a partner. And this data was collected previously by former student Justin Schaefer and postdoc Joe Spraker, but I'm in the midst of analyzing it right now, which is where this heat map on the right is coming from. But instead of diving into that today, I'm just going to skip it entirely and expose you to a different technique that you may have not heard of before. And that's RBT and seq. And this is how I'm really trying to get at the bacterial genetic requirements. RBT and seq is a transposon mutant library that you can then expose to conditions like fungal co-culture and sequence it to determine fitness contributions of individual genes. And it involves a crazy amount of plates. So TN-seq is the slightly older version of this. It's transposon insertion sequencing. You create a library of bacterial strains where each strain has an individual transposon insertion site, so it has one mutation in it. But when you have that library of strains, there should be mutations across the genome, multiple in each gene. You can then expose that library to a condition and sequence it to assess gene contribution to fitness on a genome-wide scale. But the problem with TN-seq is that you have to do pretty intensive sequencing every time you use that library. So if you want to do this when you're exposing your library to a bunch of different things, it gets very expensive and very computationally difficult quickly. So RBTN-seq simplifies the process by incorporating a random barcode that makes the downstream sequencing easier. So every transposon has a random barcode associated with it, and you just have to sequence your initial library to connect that barcode, or basically hexagon in this graphic, to a gene insertion. Then when you expose the library to a condition, you amplify the barcodes and just sequence those. So you can imagine that sequencing just those little barcodes, you can get a lot more bang for your buck than sequencing whole genomes. In the end, you actually assign a quantitative value to the fitness contribution of each gene. And unlike RNA-seq, where we're kind of reading the tea leaves based on functional annotations that may or may not be entirely accurate, the great thing about this is we don't really care about what the annotations already said. We'll know pretty specifically whether or not it matters as to whether the mutants show up in our final library. So to make this kind of a library, You start with just conjugating from a donor library, and that's what this video on the right is showing you, is me doing all of my bajillion plates to be able to get 150,000 colonies in my library so that I have an average of 1 insertion per 30 base pairs. So it's a lot of strains in one tube. From there, I'm going to sequence the library, which is what I'm in the middle of right now. So I have no data to show you, unfortunately. But then I get to start diving into experiments. So what kind of experiments do I want to do? I would like to expose the library to different conditions like nutrient stress and fungal co-culture to identify genes that are critical for life as an endohyphal bacteria. And this is really just a jumping off point, because at that point we can say, oh, these are all the things that seem to be important. Are they important across EHB? Are they conserved? What are the function of these? And this gives me something to move forward into an independent position as a theme for my future career, which I'm really thankful for about this fellowship. Long-term, understanding the genetics of these relationships will allow us to manipulate them, to learn about fungal hosts, to be able to maybe modulate pathogenicity, and even encourage plant growth promotion. So there's a lot of cool application potential for this field. With that, thanks for listening. Catch me in a breakout room or at my poster, and acknowledging the Baltras and Arnold labs, as well as the Deutschbauer lab for the constructs.
Well, thanks for that talk, Morgan. If you're interested in how the barcodes work, you know, it's not grabbing a bacteria and going boop at the checkout line. Or if you're interested in maybe on if these bacteria can make mushrooms have even more umami flavor based on what genes they make sure you go to Morgan's breakout room. Okay, our next speaker is Shio Kobayashi, and she'll be talking to us about programming T cells to do our bidding.
Sorry, I'm muted. Now I share again. Okay, good afternoon, everybody. I'm Shio Kobayashi, working in the Dr. Kuhn's lab. Thank you for giving me this great opportunity. Here, I would like to present my research overview. Okay. In 2017, the FDA approved the first chimeric antigen receptor drugs known as CAR T-cell drugs. The CAR T-cell drugs have shown a drastic result in the treatment of B-cell malignancies. However, the therapy has a difficulty in the application for the other types of the cancer or diseases. So we developed a new type of chimeric receptor to target specific T cells and to treat diseases caused by pathogenic T cells. Here, I showed our CAR T cell structure. Our CAR T cells is composed of 5 modules. One is a chimeric receptor module, and then 3D3 molecule module, and then also surrogate receptor module. Chimeric receptor module is designed to interact with TCR by MHC-TCR interaction in an antigen-specific manner. And also surrogate receptor module is designed to mimic the TCR signaling. We call this receptor 5M CAR. To explore the potential of the 5M CAR CTL treatment against diseases caused by pathogenic T cell, we tested using a type 1 diabetes mouse model. In this model, diseases occurred by the transfer of the pathogenic T cells. By treatment with 5M CAR T cells, most of recipients were prevented from the onset of the diabetes. These results indicated 5M CAR technology has potential the treatment of T1D and the diseases caused by pathogenic T cells. Based on the successful result from the T1D mouse model, I currently try to apply 5M CAR technology to the treatment of T lymphoma. These are our aims on the BIO5 postdoc fellowship grant. So now I designed a generated first generation of humanized 5M CARs Thus, I would like to show them my current progress. Our goal is applying 5M technology for clinical studies. For this goal, the design of a humanized version of 5M is necessary. As a first step, I generated HLA-DQ type 5M and investigated that the humanized version works well, like a mouse version. I transduced the humanized 5M into the mouse hybridomas with human CD3 molecules and they tested the function by IL-2 production from 5M hybridomas after the co-culture with target cells. The humanized 5M hybridomas successfully produced a large amount of the IL-2 when they were co-cultured with specific target cells, like a mouse 5M expressing cells. And it was shown that humans amplify the CAR signaling. We confirmed the function of first generation of the humanized 5M CAR, and we are moving to the next steps now. In the end, thank you for listening and thank you for big support by the BIO5 Fellowship. Thank you.
Thanks a lot for that talk, Hashio. I think that has some great implications in autoimmune diseases later, so feel free to go to her breakout session and ask her all about it after this. Our next speaker is Hui Yang, and we'll learn all about vertebrate and algae live together in perfect, I don't know if they're perfect harmony. We'll find out.
Thank you, Dr. Jensen. Yeah, we'll find out if we can truly live in harmony together. Hi, everyone. I'm super happy to be here to have this opportunity to talk about my research project. So my name is Hui Yang. I'm a postdoc in Dumel Lab here in MCB and BIO5. My research interest is a very unique symbiosis between spotted salamander and green algae. So here, this beauty is a spotted salamander, and during the breeding season, the female will lay eggs that is wrapped in a very thick egg jelly. So this is a very fresh young egg cluster. And then this in about 2 or 3 weeks, and this is how it's going to look like. So you see the eggs turns completely green because there's such a high density of algae living inside, and not just like any algae, it's a very specific species that form a one-on-one relationship, like a symbiosis with the embryo. And it will appear somewhere in the middle early stage during the embryo development. It becomes very abundant. It flourishes inside the egg capsule, and it turns the whole capsule green. In 2011, one of our collaborators, Dr. Kearney, published a paper and found something really interesting. Here, what we're looking at is the head of a developing embryo. And these red spots are the autofluorescence of chlorophyll, which means the algae does not only sitting inside the egg, but also they enter the embryo, the animal tissue, and even the cells here. This is a TM picture showing the animal cells, and the highlighted area is a algae sitting inside. Now, this makes things a lot more interesting because this kind of intracellular symbiosis is not common and definitely not reported in invertebrates. And a little bit later, another collaborator of ours, Dr. Burns, did a transcriptome study and compared the gene expression in the algae and the embryo before and after they enter the animal cell. And there are some interesting suggestive results. The algae is showing some signal that they're not exactly happy. There is a morphology change when the algae enters, and there's some stress response. As for the embryo, somehow it seems that the embryo is down-regulating the immune response to tolerate the existence of these algae. So with this, we have a lot more new questions and interesting questions coming up. So for this project, I am specifically interested in looking at 2 directions. First, about when this intracellular symbiosis the basic question is, what is happening there? Like what happens when the algae enters the animal cell? Do they still have formed this mutually beneficial relationship? Is somehow there's a material exchange between the algae and the animal? And then I also want to step back a little bit and take a look at the symbiosis inside the egg capsule and see if there's other beneficial that we didn't look into earlier, and see how having this embryo algae benefits the embryo. And right now, to do that, I'm going to go down two directions. One is to label these algae with stable isotopes, and inject those isotopes into developing embryos, and then we'll collect cells that are harboring these algae, and then we'll use something called nanosims and Raman spectroscopy. So those are basically advanced imaging technologies that can highlight those isotopes to show the distribution of these elements. So it can tell like if there are truly material biomolecule exchanges between those two between the animal and the algae. And also to see the intracellular, to see the microbial structure inside the egg capsule. Basically, the idea here is to compare the microbial community inside egg capsules between those eggs that has algae and and do not have algae, and see if the presence of algae somehow change or impacts the microbial community. So to do that, thanks to BIO5 fellowship, I get to travel a lot and do a bunch of stuff. As in right now, I'm actually in Gettysburg, Pennsylvania. We're doing gathering some of the first stage of sample collection and measurement. So this picture is actually last week. The timing is really good, so we get to get some really nice pictures seeing the female laying eggs, and we got some really nice samples. And now, right now, it's ongoing experiments to show the difference with eggs that's harboring algae and do not have algae. And then here, what my job, my work here is to carry out the experiments, and I will extract egg fluids and DNA and send them out for sequencing. And once I get back to Arizona, I will go through the sequencing data and compare the microbial structure between these different experimental groups. And then after Pennsylvania, I will soon visit another collaborator in Maine, Bigelow, and then I will do the injection experiment there. So far, we already have cell lines labeled with isotope ready, and we just need to inject them into the embryo. And once we have those samples, we will carry on do the sample preparation and move forwards towards the imaging step, which I will most likely finish when I get back to Arizona. So that's some preliminary data we got from last year. So here we can see these highlighted area is with nanosim showing us the specific elements of interest. So left here is the regular nitrogen, which showing all these different cellular structures. And on the right side, we have let's say now I'm focusing on detecting the isotopes, and we can see a specific region showing an extremely high concentration of these isotopes, showing that the labeled algae is present over there. So that's a brief walkthrough of my research, and thank you for the support of BIO5 for my trip and doing all these experiments.
I love the pictures too, man. That was great.
Thank you.
So again, if you have questions, just visit him in the breakout room after the talk. So our next speaker is Estella Jauregui, and she will be talking to us about elucidating mechanisms of sex-specific effects of phthalates.
Yeah. Hi, thank you for the introduction and thank you for the opportunity to talk to you about a project I'm working, funded by the BIO5 Fellowship. So I work with Dr. Craig. So we study endocrine disruptors. So specific in the chemical I'm focusing is the dibutyl, also known as DBP. So DBP is a plasticizer found in many consumer products. So we can find in kids' toys that are made of plastics, in the bottled waters, cosmetics. And it is known that we're exposed to 2 to 10 micrograms a kick per day through a diet of this DBP. And DBP has been known to cause reproductive toxicity. And one of the way it causes, it alters the production of reproductive hormones. So my project focused on the steroidogenic cells of the ovary and testes. So this cross section right here is a cross section of a mouse ovary. So this one right here is the follicle, and inside of it is where the oocyte is. Surrounding the oocyte is the granulosa cells, and then around the granulosa cells, it is where the steroidogenic cells of the ovary are located. So they're known as the theca cells. So their main function is to produce androgens needed for the normal reproductive hormone production. On the right side, we have the cross section of the testes, so mouse testes. These circles right here are known as the seminiferous tubules, and is where the sperm is produced. But between the tubules, we have the steroidogenic cells. This one's right here, which are known as the Natick cells. So they produce testosterone and other androgens, and their main and other reproductive hormones needed for the normal production of sperm. So any alterations in the steroidogenic cells can affect the germ cell development in the testes or in the ovary itself. So my main project for the BIO5 is to investigate the sex-specific effects of DBP on the androgen biosynthesis pathways in the steroidogenic cells of the ovary and in the testes. So in order to do that, I generated females and male Rabbit positive C17IQ positive mice, which I'm going to talk to you about next. And I'm going to treat the animals for 10 days with a vehicle control, so the corn oil, and then different dosages of DBP. So one will be the 10 mg per kg per day to represent the general population, and then the 1,000 mg per kg per day, which is just a high dose for a classical comparison. After 10 days, I'm going to euthanize the mice, connect the ovaries and testes, and then isolate the messenger RNA from the steroidogenic cells of the ovary and testes and perform RNA-seq to study the sex-specific effects of DBP in the steroidogenic cells. So how I'm going to isolate the messenger RNA from the steroidogenic cells. So I'm using this specific mouse line known as the rabbit tag. So what is important about this mouse, it has a duplicated XF4 HA tag on the ribosomal gene RPL22. So when I cross this mouse line to a Cree, so in this case, a steroidogenic cell specific Cree, the CYP17, this Cree will excite the wild type XN4 and the HA tag XN4 will be expressed. So the experimental mice will contain steroidogenic cells. So if it is in the testes or in the ovary, the steroidogenic cells will contain ribosomes with the HA tag. So using an HA antibody, I can actually I can immunoprecipitate the ribosomes with the HA tag attached to the messenger RNA. And then through immunoprecipitation, I can isolate the messenger RNA from the steroidogenic cells of the testes or the ovaries. And then what I want to do next is perform RNA-seq to study the different sex-specific effects of DBP on those steroidogenic cells of males and female mice. So what I have done so far is I have generated the male Rabbit positive C17I3 mice. So there's a cross section of the testes, and I have stained with the HA antibody, and you can see by the brown staining that the steroidogenic cells is expressing the HA tag, which means that by using the Rabbit methodology, I can actually isolate the ribosomal from those cells and extract the messenger RNA only from those cells. I also generated the female Rabbit positive C17I3 positive experimental mice. Once again, I stained the cross section of the ovary, so there's a funicle, and you can see by the brown staining that the HA tag is expressed in the theca cells, in the steroidogenic cells of the ovary. There's some in the interstitial, but mostly they're more in the theca cell. So by this methodology, I can get the messenger RNA only from the steroidogenic cells and study the different effects between the males and females caused by the DVP and see why the males usually has a greater effect than the females in the reproductive systems. So I want to thank the Crate Lab, my committee members, and specifically the BIO5 postdoc fellowships. Thank you.
Thanks for that great talk. And again, if you're interested in asking her questions, please visit her in the breakout room. So our next speaker, and we have 3 left for you all today, so please hang around, stay tuned, is Dr. Kate Rhodes. And she's going to be talking to us today about Neisseria infections and polymicrobial infections.
All right. Hi, everybody. Thanks, Michael, for that introduction. My name's Kate. I'm a postdoc in Dr. Maggie So's lab here in immunobiology. And our lab studies Neisseria biology. So we're interested in specifically Neisseria gonorrhea. And so the whole proposal that I wrote for BIO5 postdoctoral fellowship is going to be investigating Neisseria gonorrhea infection in the polymicrobial cervicovaginal environment. Okay, so Neisseria infection in the United States is still a really serious issue. As of 2019, gonorrhea is the 2nd most commonly reported infectious disease. There are about 1 million new cases of gonorrhea every year. And that's particularly concerning when we consider that we're down to 1 recommended antimicrobial class that can still treat gonorrhea. So in our field, the idea is that a better understanding of Neisseria infection biology may actually lead to novel treatment and prevention strategies that can help curb the spread of disease. So one of the facets of Neisseria infection biology that I'm particularly interested in is understanding how Neisseria gonorrhea interacts with the microbiota. So in the female reproductive tract, in healthy women, the lower female reproductive tract microbiota is predominantly composed of lactobacillus species. So these are bacteria that produce a lot of lactic acid, and they help maintain a low pH environment that assists in immunomodulation of the epithelial tissues in the cervix and vagina. Now, during bacterial vaginosis, which is perhaps the most common vaginal infection among reproductive age women, we see a shift in that microbiota population away from lactobacillus dominance towards increased microbial diversity. So we have this really diverse consortia of obligate and facultatively anaerobic bacteria. So we also see an increase in the pH during bacterial vaginosis. We see increased inflammation because some of the bacteria that have expanded in that microbiota community are potent stimulators of the immune response in the tissue. And this leads to eventual barrier damage. So bacterial vaginosis can lead to several different kinds of complications, including during pregnancy, but it also leads to increased susceptibility to sexually transmitted infections, including gonorrhea. Women with bacterial vaginosis are about twice as susceptible to gonorrhea as women with healthy microbiota. But we don't really understand why. So the goal of this BIO5 fellowship is to try to get at what is the mechanistic link between bacterial vaginosis and NGO infection. Right, so to do this, I'll be working very closely with Dr. Melissa Hertz-Krabelitz and her team at UA Med Phoenix. So the Herbst-Kralevitz lab has figured out a really cool way of studying the pathogenesis of bacterial vaginosis in vitro. And it starts with the creation of this, this 3D tissue mimetic. And Rob gave a really great introduction to this process, so I won't go into much detail here. But the first step for the pilot experiments I'll be running with Dr. Herbst-Kralevitz and her lab is to generate these 3D cervical aggregates. So we'll be taking human cervical cell lines and growing them in a rotating wall vessel bioreactor, which encourages these cells to aggregate and differentiate into tissue that very closely resembles a cervical tissue in vivo. So the next step is to take those cervical aggregates and colonize them with one of a few different cocktails or synthetic cocktails of representative microbiota species. So these are different mixtures of bacteria that have been cultured and characterized by the Herbst-Kralevitz lab. And each one represents a microbiota at a different stage along the progression of health to bacterial vaginosis. So once these cervical aggregates have been colonized with our different community types, I'll then be infecting with Neisseria gonorrhea. And then at different time points, I'll be taking samples from these different communities and then assaying them to look at the ability of Neisseria gonorrhea to survive in these environments and its ability to actually infect. I'll also be banking RNA and supernatant from each sample for later analysis. So the goal here, really with this pilot experiment, is to establish baseline conditions for studying Neisseria gonorrhea infection in this 3D community model, which we hope to use for many other experiments down the line. And also the material that we're generating, so the RNA and supernatant, will go for RNA-seq analysis that we'll be using to examine Neisseria gonorrhea gene expression under each condition, and metabolome analysis to look at changes in the metabolites produced in NGO-permissive versus non-permissive communities. And the hope of all of this is that we can hopefully identify key Neisseria pathways and community-derived metabolites that are responsible for this BV-NGO co-infection. So once we're able to establish this mechanistic model, we'll need to confirm everything through NGO knockout construction and testing. And then hopefully we can expand and refine on this concept using additional community cocktails with new members incorporated from clinical samples, as well as looking into the contribution of some pre-established Neisseria gonorrhea host interaction factors using mutants in those, as well as immunoproteome analysis. So I'll be happy to take any questions during the breakout session, but first I would just want to say thank you to all of the people that are helping to make this project happen, my funding sources, the BIO5 postdoctoral fellowship, all of the members of my mentorship committee, and then the SO lab and the Herbst-Kralevitz lab. So thank you.
Awesome, Kate. Thank you very much for that. We actually skipped into Michael Plank, and he's going to be talking to us about transient and sustained targets of protein kinase A in the transition to rapid growth. So we'll find out what that rapid growth is of.
Okay, great. You hear me and you see my screen?
Yes.
Okay, awesome. Okay, so I'm Michael. I'm a postdoc in Andrew Capaldi's lab, and I'm going to tell you briefly about a project where we're looking at the kinetics of protein kinase A signaling. So protein kinase A, PKA, is actually a kinase that has been studied for a very long time. It's the second kinase that has been discovered. And so people have been studying it for 50 to 60 years. And if you talk to somebody, then they would tell you PKA signaling is pretty well understood, at least in its basics. And a typical way in which you would study kinase signaling is you take a condition where you know that the kinase is active and a condition where you know that it's not active, and then you would look which proteins are phosphorylated in these conditions, and use this one as a starting point, what this kinase may be doing. So I want to challenge this notion that protein kinase A signaling is well understood a bit here. And the reason why I want to challenge it is because I think we're thinking too simplistic about this. So a statement that you might hear in protein kinase A signaling or any kind of other kinase signaling may be something like, when kinase X is activated, then it phosphorylates its targets Y and Z. And as I said, I believe this is often too simplistic. But let's go back to pKa. So pKa in yeast is active when the yeast cultures have glucose, and it's not active, or at least very little active, when those cultures don't have glucose. So the Capaldi lab where I'm working is actually a lab that's interested in TOR signaling. And the reason why they got interested in pKa signaling is because both TOR and pKa are the major growth regulatory pathways in budding yeast. So they did a study where they asked in which case which kinase is more important. And what they found very interestingly is that TOR signaling is very important once cells have reached their new steady state. So they have been in glucose for a long time, and then TOR is driving this growth. Versus pKa is very important during the transition. So when you go from a state where the cells don't have glucose to the state where the cells will have glucose. At the same time, I was working in my last postdoc doing phosphoproteomics, whereas those data from the Capaldi lab are transcriptomics data. So I was doing phosphoproteomics, and I was one of these people saying, okay, we want to find out about a kinase by looking at the state where the kinase is not active, i.e. in not having glucose, versus where it's active in having glucose. And so hearing about this data from the Capaldi lab, I thought this would be a great opportunity to join forces, and I joined the lab, and we were using the expertise of the Capaldi lab in this transition and my expertise in phosphoproteomics. And actually in hindsight, it's a pretty obvious thing that you would want to look at pKa signaling during this transition, because it has been known for a very long time that there is a spike in cAMP, which is the second messenger that activates pKa during this transition. And then afterwards it goes to a lower level, which is still higher than initially when the cells didn't have glucose, but it's once they have achieved this new steady state in glucose, this level is lower than during the transition itself. So as I said, what we did is doing phosphoproteomics during this transition. And we indeed found about 50 targets that are affected by pKa signaling, many of them that hadn't been reported before. And how can we say that these are pKa dependent targets? Well, so what we did here in this phosphoproteomics experiment after glucose addition to the cultures is we either added an inhibitor of pKa or we didn't. So obviously those sites that respond if there is no pKa inhibitor around, but they don't respond in the presence of the inhibitor, those ones are the pKa dependent sites. And as I said, we found around 50 or so such targets, and many of them new ones. And the second interesting thing is that we noted these very different clusters in terms of their kinetic behavior. So what you see in the top is the ones that show a sustained behavior, where the phosphorylation stays on, even so the pKa activity is reduced again at these long time points. And in the lower cluster, these are the ones where we mainly found many new targets, are the ones that spike in their phosphorylation, but then go down again. And in red, these points would be where we inhibited pKa, so this we don't see an increase here. So based on these data, you can obviously ask, what is the biological relevance of these different kinetics? And this is something that I'm actively working on and that I'm not going to talk about here. The other question that you want to ask is, how are these different kinetic behaviors achieved? And I'm going to talk about this in a nutshell, and then I hope if you're interested, you'll have time to go back to the poster and have a look at that. So the first question that we asked is, are these kinetics determined by pKa itself or by something independent, like for example, the phosphatase that's acting on these sites? And what we did here is we looked at the correlation between the kinetics and the susceptibility of those sites to be affected by the inhibition of pKa. And we found that there's actually a pretty good correlation, not a completely straight line, but a good correlation, which means that in many cases, this kinetics is dependent on the substrate quality of these sites for pKa itself. We then looked back at our phosphoproteomics data, and what we found is that the targets that are very sustained are actually the ones that have a very strong pKa motif. So RRXS is a very strong motif around the phosphorylation sites, whereas the ones that are not sustained or much less sustained are the ones that have weaker pKa motifs, which again goes back to this idea that it's the strengths of substrate quality itself that determines these kinetics. and not some independent factors, and especially that this strength is encoded in the sequence around the phosphorylation site. And finally, to drive this idea further home, that it's the sequence around the phosphosites that determines the substrate quality and therefore the kinetics. We did some in vitro assays where we took peptides that represent the linear sequence around the phosphosite. And again, what we see is that those targets that represent very sustained targets, they are the ones that are strong substrates in vitro, versus the ones that are these transient targets, they are not good substrates in vitro. So if we go back to this very simple statement from the beginning, when kinase X is activated, then it phosphorylates its targets Y and C. Then you can see that there may be problems with this. On the one hand side saying, well, kinase X is activated, but maybe the kinase is not just on or off. Maybe it's in a state where it's very strongly on during the transition, and then later on, when the new state is reached, maybe it's partially on. And the second part, it phosphorylates Y and C. Well, maybe it phosphorylates Y and C during the transition where it's very highly active, but when it's less active, then maybe it only phosphorylates one of these sites. And on the poster, if you get a chance to go back, we're discussing a bit that this is actually only the second case in which it has been shown that substrate quality determines the kinetics of phosphorylation of targets of a kinase. And with this, I would like to thank also the BIO5 for the postdoctoral fellowship, and thank you guys for listening.
All right, thank you very much, Michael, for that great talk. Nitesh Khandewal, and if you've ever, if you've been looking at the news and you've seen these really gorgeous structures of the spike protein from SARS-CoV-2, a lot of them are done by this technique called cryo-EM. So I think, Nitesh, you're going to tell us a little bit about cryo-EM and what we can find out from it here.
Thank you, Elena, for sharing my presentation. And I'm sorry, guys, you guys have to wait because of me. I'm postdoc in Tomasiak lab, and I work on a structural determination of yeast vacuolar ABC transporter. Next slide, please. So ABC transporters are mainly present on the plasma membranes of different organelles. They compose of two parts. One is transmembrane domain, another is nucleotide binding domain. Transmembrane domain, they usually bind to the substrate, and nucleotide binding domain, they are like powerhouse for ABC transporter protein. Like they use the energy, use the ATP, and change the structural, make these structural conformations and transport the substrate out. So why we are interested, next please. Okay, so why we are interested in ABC transporters? Because they are present in prokaryote, eukaryote, like throughout the different phyla. This is one of the largest family. But the interesting part is ABC transporters, they are involved in multiple functions. Like in case of like a normal cell function level, like they maintain the lipid homeostasis, they maintain the ion transport and homeostasis, and like different kind of substrate range, each wide range of substrate. So for case of like pathogenic bacteria, they involve in nutrient uptake and secretion of toxins. But one of the most attention they got is by they provide drug resistance in different phyla and different organisms. And this called like multi-drug resistance phenomena, because one transporter sometimes can provide drug resistance to multiple kind of drugs, which are structurally or like functionally different way. So these multi-drug resistance can be seen in human when during the cancer, like during chemotherapy. They can be seen in fungus during like different kind of fungal drug resistance, like in Candida albicans and like in Cryptococcus, and as well as in bacteria like tuberculosis. So these transporters get the attention a lot. And next, please. So usually these transporters have 7 transporter subfamily, and we are interested in ABCC subfamily, which is out of these 7, one of the subfamily. So one of the interesting point about ABCC subfamily is they have one extra transmembrane domain, which is called as TMD0. And the second interesting thing about this transporter family is they have one of the extra structurally undetermined region, because it's like very floppy region, but this region get phosphorylated and differentiate and regulate the activity of this transporter. So most of the ABCC family transporter in yeast are present on the vacuolar lumen, and they can transport different kind of substrate into the vacuole. Next, please. So in Tomasiak lab, I'm working on two transporters from two different organisms. One is YCF1 from Saccharomyces cerevisiae, and another one is MLT1 from pathogenic fungus Candida albicans. Next, please. So usually YCF1 and this transporter do the conventional function. Conventional function means because the name suggests transport. So they transport different kind of substrate, like they transport different heavy metal, they transport glutathione, which cause the oxidative stress, different kind of bilirubin, and like other than that, they also involve in non-conventional function, like they present on the vacuole, they involve in vacuole fusion, they take over the, like they help the cell to survive from oxidative stress. Next, please. Similarly, MLT1, which is homologue of YCF1 in Candida albicans, it also involved in, not only involved in like ion transport, it involved in drug transport as well. So in my PhD during I able to find out like the mechanism of MLT1 in drug transport. And continuation of my PhD in Tomasiak lab, I was interested like how these transporters function and what is the structural basis of determination of this different kind of activity and their regulation. Next, please. So today I'm going to talk about one of the transporter out of these two, which is YCF1. So in YCF1, it's from Saccharomyces cerevisiae. So we express this transporter in Saccharomyces cerevisiae. Next, please. So there is like we use this construct to express the YCF1 where we have N-terminal flag tag, C-terminal his tag, and we can use two different kind of tag to purification. Using these tags, I have discussed in my poster session, like we able to purify this protein. One is wild type and one is mutant version. Next, please. So after purification of this membrane protein, one of the important thing is these are active outside of the cells. So for that purpose, we tested this activity, and which is like, because they are like name suggested, ABC transporter, ATP binding cassette transporter, they consume the ATP. So we check the ATPase activity of these transporters. And if you can see YCF1, this is the wild type transporter, and second one, the mutant version, which is E2Q. So YCF1 was highly active, and E2Q mutant was a mutation in the nucleotide binding domain 2 in Walker B. And it's well known and conserved in among ABC transporter. Like if we mutate this residue in E to Q glutathione, E to Q, the transporters become inactive. So we see the similar thing here. Now, after we are satisfied our protein quality is good, it's active. We wanted to know like we wanted to like explore the structural biology of this protein. And for that purpose, we use cryo-EM technique, which is one of the like hot like in these days, because in 2017 you all know like we get the cryo-EM technique got the Nobel Prize in chemistry for developing this technique where we can study the biomolecules in the like in vitro phase or solution phase. So using the cryo-EM technique, next slide please. Using the cryo-EM technique, we could able to solve two structures of YCF1. These are two different conformations. If you can see here, this is the inward facing conformation. Both conformation, they are facing inward facing, like the cavity is open towards the inside. Okay, and they are like inward facing wide versus narrow. We say that inward facing wide versus narrow because of the distance between these two NBD. So this is like wide and this is narrow. Interestingly, with this with our map, we could able to build all the site of like TMD0, TMD1, TMD2, as well as the R domain. So far, there's one structure of ABCC transporter available in human, which is CFTR. And there, one of the biggest trouble was they could not able to solve the R domain structure because it is very floppy. And they are like very conserved. So we could able to solve this R domain structure, which we are very interested, because not only that thing, in our structure, we also find that there are 3 sites which are phosphorylated, and we could see those in our structure as well as in our mass spec data. So next slide, please. So here, these two structures are overlapping, the overlapping view of these two structures and this R domain we are highlighted, in which you can see 1, 2, and 3, these 3 phosphorylation sites in both structure 1, 2, and 3. So that was one of the big highlight, because we could also able to show how this phosphorylation regulate this transporter activity, because if we mutate single residue of this phosphorylation site, the activity is lost. And can you click on this video? So this is just a view like how these two conformation, it's very fast video. So you can see how this protein changing from one conformation into second conformation. These are shown in different angles. And this is like bottom angle. You can see the movement is very high in this bottom angle, and in the top, which is present on the membrane, the movement is very low. So anyways, like, so so far we have two conformation. One is wide and narrow, but both are apo. What we are interested in to dissect the more like different conformation, which is like ATP bound and substrate bound conformation. Next slide, please. Next slide, please. Sorry. Yeah. So with this, but the other details about this I have discussed in my poster. But I just want to give it like this home, like take home message. Like we have two conformation of YCF1, and we could able to show how this phosphorylation sites they dependent regulation of this transporter. And I would like to acknowledge where I collect the data, like PNCC. So far, I collect the data for my cryo-EM sample at remotely. I prepare the sample in the lab and send it to the PNCC center. With the BIO5 postdoctoral fellowship, I want to learn this technique because it's a very costly instrument and $10 million instrument. And NIH centers, they provide you, the people who are interested, they provide a 15 days like training session. So with the BIO5 postdoctoral fellowship, I'm going to attend a session personally, which is so far because of the COVID time, is the date is not finalized. But my future aim is with this BIO5 postdoctoral fellowship, I want to learn to handle this instrument itself. With this, thank you. I would like to, next slide. I would like to thank you everyone. And I'm so sorry with this all technical issues.
Hey, it happens. Don't worry one bit. Thank you so much for sticking with us. Thanks for being patient with us. Thanks for duking it out in the test so we could hear you talk. That was, because that takes a lot, that takes a lot too to kind of stick around and do that. So we appreciate you as well. So with that, we're happy to now take some questions, but for those questions, we will ask you to go to the breakout rooms. So in addition to that, what we'll also say is that the posters will be up until 5 o'clock today. If there was like one crucial detail that, you maybe didn't want to ask in the question, but feel free to still ask that question. But you just couldn't get enough of the talk. You wanted more. Please visit the posters, but also please visit our speakers in the breakout session. So I'll give them a chance to go to their particular breakout rooms, and you all are also welcome to go to the breakout rooms if you want. And we'll see. I think we'll start seeing some of our speakers disappear once that is able to happen. Or maybe not. Okay, the rooms are now open. Yep, some of our speakers are disappearing. So feel free if you want to just leave now, that's fine. But if you want to go to the breakout rooms to visit our speakers, you can do that as well. But yeah, they are now open. And for those of you who are left, I'll say have a great rest of your afternoon.